Hello everyone, I am Vinoy Krishna and I teach the GS Science and Technology as well as a portion of the optional paper Anthropology here at Vajiram and Ravi. I am meeting you today particularly with respect to the science and technology part of the general studies, wherein my prime focus in this brief discussion that we are going to have is how to build the fundamental with respect to the multiple current development understandings. We would enter into an area of life science with relevance to the biotechnological applications. And we would see how just the understanding of a basic concept in that could amplify your understanding of multiple current developments that you are coming across in the different media. All of you, I am assuming at this point, are already aware about every living cell having the blueprint of its functions in the biomolecule called genetic materials. People, this genetic material is stored within a cell, primarily inside the nucleus, as a DNA packaged in the form of chromosomes. So chromosome is nothing but the packaged form of the thread called DNA. The concept called genome that you have recently also come across in the newspapers with respect to the Genome India project is, on a plain understanding, means the sum total of the entire genetic material present in one set of chromosome. When you know that a human cell in its nucleus has 23 pairs of chromosome, meaning every chromosome with a double copy, you understand that it has two sets of the genetic material or the chromosomes, meaning two genome sets are present in every human cell, except for the gamete cells called the sperm in males and ovum in females. We will not enter into that for the moment. बच्चों, the simplified most presentation of the life at the cellular level happening across the biological world is this. Present inside the cell, within the nucleus, is the genetic material in the form of DNA. DNA is a double-threaded or double-stranded molecule having the information in the language of four nitrogenous bases A, G, C and T, which should be assumed as the four letters in the language of life or four alphabets in the language of life. You see, the segments of the DNA which code for a specific protein is called as gene. In a human genome or the entire genetic material present in a human cell, we have an approximate 20,000 different genes, each coding for a separate protein. The very fundamental of life is any gene which needs to produce the corresponding protein will first create its own photocopy in the form of an RNA, which is a single-stranded nucleic acid called mRNA or messenger RNA. This mRNA, which is the photocopy of the gene which is expressing itself, and hence the term you will come across called gene expression, will then pass out of the nucleus into the cytoplasm of the cell, the fluid present outside the nucleus, people. This mRNA is immediately then anchored on by or latched on to by the ribosomes, the specific particles which are floating around in this medium within the cell. It slides over this mRNA and in doing that gets the message that the mRNA is bringing from the gene read and then converted into another extremely important biomolecule called the protein. People, the protein is a polymer of amino acids. There are almost 20 different standard amino acids using which you may create infinite number of proteins. And it is these protein, people, which ultimately facilitate the expression of characters. If you wish to understand it through an analogy, the entire genetic material could be understood as the script. For example, the constitution of a country, telling how to govern it. And a particular gene might refer to a particular article. And that gene's photocopy in the form of mRNA ultimately gets converted into the protein. The proteins in your body are very much like the executives that we have for a country. Now, my main focus would begin here onwards. Most of you must be already knowing this, called as the central dogma of life. A dogma because it is a belief that is not open to debate or change. Across the living world, every cell of every organism does the same. But then begins the understanding. What happens to these proteins which are created from a gene? People, this protein can have multiple fates, or you can understand addresses or destinations. One, the protein could be produced and required within the cell itself by the cell, called intracellular proteins. Two, the protein produced could be, right after the production, be thrown out of the cell to travel through your blood circulation to reach to the different parts of the body, called extracellular or secretory proteins. Very much like your insulin, a hormone which is proteinaceous produced by the beta cells of the pancreas, or the antibodies which you might have read the B cells produce after production are thrown out into the circulation. But the most interesting is the case of those few proteins which are not clear whether they are required within the cell or to be thrown outside. They are, in a literal sense, the fence sitters. People, these proteins, after produced, the third category that I am talking about, might get produced and may get tacked on to the membrane like a pin does to the pin cushion. You see, I am drawing these proteins in different shapes. Say for example, like a pin. And then here with a cup. And then here, more like a gate. Each of these proteins, people may have different roles. Listen carefully. Let us say this first category. There is a group of membrane proteins which behave as the surface markers or surface antigen. But what do we mean by the marker? Marker is anything using which you can identify something. Let's say the house number written on the outer face of the building or the name of the owner. That is the marker, who the house belongs to. There are different types of these surface proteins acting as the surface marker at different levels. See how. Some of the proteins are unique to a specific cell of the body or tissue of the body. Let's say the heart cell, lung cell, liver cell, bone cell, blood cell, based on what specific functional type the cell belongs to, it will have some unique surface protein on it. So do the cancer cells. Every cancer cell might also have some specific identity proteins. Keep it in your mind, we'll see how in a recent development this particular aspect could be utilized to understand. So using this we can identify the specific cell within your body. Further, we have some specific surface proteins which act as the marker of an individual, or it is unique to an individual within a species. People, what it means? If me as a member of Homo sapiens species have a particular protein present in all my cells, that protein would be unique to my body, and everyone else will have their own unique surface protein specific to that individual person. And you see how this will give you the easy understanding whenever we go for transplant of the tissue or an organ from a different person, which structurally and functionally looks exactly like yours, still your body may be intolerant and not receive it. A phenomenon that you all already know as what you call tissue rejection. How? People, our immune system works on a very basic principle. The principle of discriminating between self and non-self, and be absolutely intolerant towards non-self. So how would it identify the tissue which is getting transferred in your system is not originally belonging to you? Obviously, through that specific individual specific surface proteins. It would be good if you remember that a particular category of proteins which are specific to every individual is called HLA protein. UPSC could ask you this. Human leukocyte antigen. That's individual specific. And what kind of a protein is it? Membrane protein. And third, people, there are specific surface markers which are unique to a species, which means all of us, by the virtue of belonging to Homo sapiens as the species, would have specific proteins which are common to all of us. But it would be uncommon to some other species. And it is using these proteins that your immunity would again try to revolt if you try to import a tissue from a different species. This understanding would help you ask a question and understand better a news related to xenotransplantation, something which has been quite popular over the last 2-3 years. Chance of getting reflected in the question paper. Xenotransplantation. Xeno is for stranger. Strange here is the species. You must have all read about the attempts at importing the organs grown in pig's body for the humans. How would we do that? It would require genetic manipulation of the pig. To do what? You would remove those very genes from the pig's cells which are known for coding those proteins which are unique to the pig. If you do that, the pig's cell will not have the marks of identity. It would become easy to fool the human's body in accepting it as its own. Although that is not the only change you need much more. That's a matter of detailed discussion. But then you see that the xenotransplantation is possible only if you rob away the mark of identity from the different species you are importing the tissue from. And this would make it possible for you now to easily understand the news on xenotransplantation. Right? This is a very recent news where the kidney which is grown in the pig's body has been transplanted into a living person. The earlier attempts were done on a comatose fellow, the person who was in coma. This is for the first time done on a living person. The first step would be, as I told you, to remove that surface protein by knocking the gene responsible for it out. Now let us take it further. So you have understood how for xenotransplantation that surface marker removal is important. Further, people, can you see the second group of protein? You can see by the shape itself, it is easy to identify this as a receptor protein because it is shaped in such a manner that it has a pocket onto which something can come and bind if it has got the right structure, just as the right key fits into the right lock hole. This is called as the receptor protein. Now if I club both these concepts, receptor and the surface antigen, let us see how you can easily understand the recent development, which, take it from me, would stand a good chance of getting reflected in your prelims and mains both. What we call as CAR T cell therapy. People, it refers to chimeric antigen receptor, T cell. You see, T cells are a specific type of immune cells. Also, better known as T lymphocytes. You might be knowing that these T cells are involved in providing cell-mediated immunity. Anyway, the term chimeric means fictional, imaginative, not natural. How about an animated character, a horse which can fly, has wings? Imaginative, chimeric. So what are we producing? Something which is not natural. People, a T cell. T cell having what? T cell having a specific receptor. Receptor against who? A specific antigen. And you see how the CAR T cell therapy is revolutionizing the world of cancer therapy and has been in the news particularly because the CAR T cell therapy method has for the first time got the Central Drug Standard Control Organization CDSCO approval for treating leukemia and lymphoma, the two related forms of cancers. You see how? In a quick manner, I'll just try to explain. The person suffering with leukemia, the blood cancer, you draw the T cells out from the blood, and like any other cell, this T cell also has its own genetic material. But we have to make this T cell chimeric, something which is not natural. What we do, people, is we use a suitable vector. Vector is a carrier. Normally for animal cells, adenovirus is a suitable vector because it has the ability to infect the host animal cells and get its own genetic material integrated with that of the host. In this case, T cell would be that host. Let's say this adenovirus carries a particular gene. For the time being, you have to know gene as a segment of DNA that codes for a specific type of protein. If we successfully get this adenovirus to transfer this gene into the T cell, the red shaded area is supposed to mean that the transfer has happened. This T cell is called recombinant T cell. Now, if this T cell expresses that gene, what did I tell you? Gene expression means the gene producing the protein it has got the information for. People, the specific protein that this foreign gene codes for is a membrane protein out of the three fates that I had told about. So it will immediately go and get itself tagged on the membrane. And by the shape itself you can see it is a receptor protein, meaning it has a pocket that can bind to something. Now your T cells have been suitably manipulated to act as a very aggressive T cell. But what is still a secret? Against what is this receptor? What does it bind to? Let us check out. This T cell, people, is first made to multiply, copy into multiple number by a method called cell culture. You put this cell into a proper medium wherein it will keep dividing itself. But your cell culture is a very simple thing. Every day you prepare curd at home, you add the little bacteria into the warm milk, the bacterial cells use the milk as nutrient and multiply their number. That is nothing but bacterial cell culture. Similarly, you will grow these T cells into multiple copy numbers. So what you now have is such CAR T cells army, and this army of many such receptor expressing cells introduced back into the suffering individual, leukemia patient. And you know what? Within the person's body, you already know that the cancer cells are present. And back then I told you, every cell has got the specific, every specific cell has got a unique surface marker or an antigen. So do the cancer cells. So these cancer cells, they have the specific antigen or the protein on the surface, people, called CD19 antigen. And I think you can now connect the dots easily. The T cells that you have formulated, they are made to have the receptors against these very antigens. So your T cells are now made to be a very precise and aggressive killers of the cancer cells. They'll find them out and kill them. This CAR T cell method, which is seen as a revolution, has been successfully tried across the world, and India fortunately is also not lying behind, wherein in an experiment or a trial that started as a collaborative mode by DBT, IIT Bombay and Tata Memorial Hospital, has come to a point where the first startup which got incubated at IIT Bombay by the name ImmunoAct would be important. And the drug formulation, NexCAR19, 19 for CD19, has got the CDSCO approval. And the chances of it getting asked becomes even brighter because we are already getting to have the success in treatment getting achieved. People, the first patient free of cancer, indigenous CAR T cell therapy brings treatment cost down from rupees 4 crores, 3 to 4 crores was the initial cost at the time of the trials, down to 40 lakhs. Dr. Colonel V.K. Gupta is the first person to have received the benefit. From the same fundamental of that central dogma that I talked about, gene expressing the protein, people, and the protein then getting decided to go on the membrane, there is another recent development that you can find an easy understanding of. That of mRNA vaccine. Let's see how this understanding becomes a cakewalk with this as a template. People, what is a vaccine? Vaccine is any entity. It could be anything. I mean, you have studied so many variants. Attenuated or live virus or bacteria, subunit, the DNA or the mRNA, which when introduced into your body generates immunological memory. Immunological memory. That is, you will have the memory B cells, T cells created without giving the disease. That is the smartness in this. How normally you develop immunity? By getting the disease, fighting it out, wearing immunity as a medallion that you have earned in that fight, right? But vaccine, people, is rather a cheat code wherein you fool your body into developing immunological memory without causing the disease. There are multiple ways to do that. Vaccine deserves a lot of respect for being the single scientific innovation that has saved the maximum number of human lives. And in that, mRNA vaccine could be treated as the single biggest revolution. You see how? We will take the example of something which has been most popular, the attempts at developing COVID vaccine. Listen carefully. You call the coronavirus by the name because the virus has got a crown appearance, resplendent with these proteins, more like the jewel on the head of this corona. Without a doubt, corona ruled over us. People, these surface protein, you see surface protein, present on the coronavirus are called spike protein. Spike protein. And these spike proteins are the ones which are the most identifiable feature of the coronavirus. Obviously, a protein must be coded by some gene, and hence the photocopy of the gene, the mRNA. So how we devised a brilliant method of making the vaccines against COVID very quickly and very efficiently. You see, I've already told you that adenovirus is one of the most preferred vector carrier of a foreign substance within an animal cell. Let's say an adenovirus is packaged with the mRNA. mRNA coding for what? That's obvious. The spike protein. The spike protein. And let me also tell you, we stand at a point today wherein it is possible to produce any RNA or DNA of your choice sequence in the lab. So we can easily lab produce any mRNA of our choice. So that's not a problem. So you just need to formulate that mRNA in the lab, package it under the adenovirus. Your vaccine is ready. It is this which is given to the person as intramuscular injection. And see how it does the wonder. People, let's say what I'm drawing here is a spindle-shaped muscle cell of your body. Since this adenoviral formulation is given to you as injection, imagine the virus goes and fuses with your cell, muscle cell. Let's say this is the nucleus of the muscle cells with the DNA inside. And as it fuses, it introduces the mRNA. And what happens to the mRNA? You have already learnt in the central dogma. What happens to the mRNA, people? This mRNA will be translated by the ribosomes. They produce the protein. They read the message and convert it into the protein. So obviously, this mRNA here would also, within your muscle cells, be translated by the ribosome of the muscle cell to produce the protein. I already taught you, every protein produced in the cell has already got a fate, either required within the cell or to be thrown out of the cell, or it will go and bound on the membrane. Since you already know, the mRNA was coding for spike protein, and the spike protein is a membrane protein. This protein would immediately go and get itself tagged on your own muscle cells. And then you already know the basis of immunity. What is that? The ability to discriminate between self and non-self. This spike protein, even if it is produced in your own cell, will come across to your immune system as non-self. It has never seen it within you. And hence, this non-self protein would be sufficient to stimulate your immune system and generate immunological memory. But do you understand? The spike protein in itself is not capable of causing the disease. It requires the virus. So you got the immunological memory sans the disease. It is like fooling your immune system. The moment it sees anything with the semblance of a virus, it will not take a risk, it will prepare. You must have seen some mischievous souls making up the reels wherein the sleeping dog and they put the tiger, soft toy, behind it. The moment it turns, it does not waste time to check whether it is real or not. It springs 10 meters away. Exactly what you do to your immune system. There are many other examples of how this very same fundamental people of within a cell, how the different genes coding for mRNA and then protein and then expressing it in the different manner could lead you to the understanding of many current developments. But I leave you with few more easy concepts that could be helpful for your prelims in particular. Some definitional terms. Just as we understood the total genetic material present in one set of chromosome is called genome. And you know, within a genome there might be a total of nearly 20,000 genes. Not all the genes in all the cells of your body are expressing. For all the genes which are expressing, they are producing the RNA, just as the sum total of all the genetic material is called genome. Just as the sum total of all the genetic material is called the genome, the sum total of all the RNAs produced within a cell is called transcriptome. Please remember, sum total of all the proteins produced within the cell, there are innumerable number of proteins created. All the proteins considered together make up what you call the proteome. And then people, your entire body at every time is having multiple biochemical reactions happening as long as we survive. Metabolism. The product of all these metabolic reactions can be called as metabolites. And the sum total of all the products or metabolites can be called metabolome. Remember these terms. And quickly work out if the study of genome, the total genetic material and its sequence, mutations, everything is called genomics. The study of the transcriptome will be called transcriptomics. The study of the entire set of proteome will be called proteomics. Metabolome will be called metabolomics. And people, all these genomics, transcriptomics, proteomics, metabolomics are the good windows to look into the health status of an organism. They are all studied together in a more holistic way and are better called integrative omics. Expect UPSC any day to ask on these terms itself. There was once a question on transcriptome. What is it? The leaving message is, whenever you study science and technology, it comes with one challenge, that it is horizonless. Every year you will have multiple current developments to read about. The moment you reduce the concept into some basic fundamental understanding, you can easily create the extensions of understanding and retain it better as well. From the very same concept, we can have multiple other applications also which are coming in the news that could be understood. That for some other day. Stay motivated, keep working hard. Thank you.